Have you ever been curious about what goes on in someone's head? Have you ever wondered what it might be like to see inside of a brain? Or maybe learn how different parts of the brain react to the world around us? Neuroscientists have. Being able to see into a translucent brain not only looks cool, but provides practical and valuable information in 3 dimensions about many things important to researchers, from what areas of a brain are active under certain conditions to the winding path of a single neuron projecting across the brain. Let's take a few minutes to explore the clearly awesome technique of tissue clearing.
Tissue clearing refers to a chemical technique used to create translucent tissue, often paired with another labeling technique to visualize different cell types. Surprisingly, clear tissue is older than you think. The first scientist to successfully create clear tissue was Werner Spatzels in 1911. This process was improved upon over the years by scientists studying ears, embryos, and even hearts, and in 2007, Hans Ulrich Dolt combined tissue clearing with light sheet imaging to detect single neurons in a mouse brain. Tissue clearing works by removing or altering proteins, lipids, and other light-absorbing molecules so that small fluorescent markers are more visible. This process limits the scattering of light found with opaque tissue and allows scientists to get a deeper view of intact, not sliced, tissue. The basic steps of tissue clearing are fixation or preservation of the tissue, labeling the cells with a fluorescent marker or tag, the removal of water and lipids from the tissue, and refractive index matching, a process that matches the refraction of the tissue with the refraction of the surrounding liquid or gel it's placed in so that less light is lost or scattered, providing a clearer image. The 3 main categories of tissue clearing techniques are hydrophobic, hydrophilic, and hydrogel-based processes, each with their own unique benefits and drawbacks. Hydrophobic methods tend to be faster, but also run the risk of bleaching some of the fluorescent molecules. Hydrophilic methods tend to be safer, but often take longer than hydrophobic methods. Hydrogel-based methods allow for RNA imaging, but can often expand the tissue size, which could be good or bad depending on your output, and could require different equipment for processing.
Light sheet imaging is one of the most widely used imaging techniques for cleared tissue, especially in neuroscience. This technique combines relatively fast imaging with less risk of photobleaching and has the added benefit of allowing one to view tissue in 3D. The tissue is illuminated by lasers that form a wide band, or sheet, of light, and a series of lenses and filters amplify only the signal of interest while blocking the rest. The tissue can be rotated and imaged from all angles, and these images are processed to create a 3D image of the tissue. In light sheet imaging, photobleaching is minimized. Sections are imaged in a clear, crisp manner, and imaging is often faster than with other techniques.
And now, let's take a quick look at a 3-dimensional image of a cleared mouse brain, focusing on the CA2 region of the hippocampus.
The possibilities for this technique in neuroscience are limitless. Some of the cool things that researchers are doing now include using virtual reality to not only walk through the brain, but to also visualize and trace the projections from a single neuron from its origin to its end. This could provide researchers a new way to see how neuronal projections in one area of the brain change with different experimental conditions. It's clear, brain clearing is the future.
Were in the way. We'll use the art to see in 3D space. Neurons with bright fluorescent scenes. We're looking at bright